function, structure of that gene or protein, then location, cellular and chromosomal, clinical effects of mutation, as well as similarities of biological sequences and their structure. So if you consider, I gave you last time the example of amylase example. So what amylase regard information regarding amylase will be present in biological database. So amylase is first of all the gene which is expressing amylase enzyme. So sequence of that gene will be present. Now amylase, if you consider amylase from E. coli, so E. coli normal temperature is, what is the normal growth condition for E. coli? Can anyone tell temperature? Can anyone tell optimum temperature for growth of E. coli? Vishakha?
फोर्टी डिग्री सेल्सियस.
हाँ
40 डिग्री.
40 डिग्री नहीं है. Other cases where E. coli is present, can anyone tell? It is normal gut flora or intestine. Human body मध्ये intestine मध्ये हा E. coli is present. And what is the function? It is symbiotic association. Humans will provide protection food to E. coli, and E. coli will produce vitamins. त्यामुळे आपल्या फिसेस मधनं आहे की नाही, इट इज नॉर्मल फ्लोरा फॉर फिसेस. इट इज सिक्रेटेड थ्रू excreta, human excreta. And when it is there in intestine, it is beneficial. But if it will go in blood or lung or any other part, it causes diseases. So what is human body temperature?
37.
So optimum temperature of E. coli is 37. बरोबर का? बरोबर ना? It is 37 degrees Celsius. So the amylase which will express the amylase gene, which will express the amylase enzyme, it is optimum temperature is 37 degrees Celsius. Then for example, there is a bacteria, say bacillus, which is cryophilic, means grows at minus degree Celsius, minus 10 degrees Celsius, found in, for example, say Himalayas, under the ice. So there also it will produce amylase, but the amylase activity is maximum at sub 0 degrees Celsius. Both the genes are there, so what it will contain? Then amylase from human, the sequence is in different little bit, but the function is same, to degrade starch. So biological database is what it will contain. It will contain nucleotide sequence from various sources. Barabar? Then second part comes structure. Now structure, it may be of mRNA or protein. So as the nucleotide sequence is different, not higher percentage, but 10 to 15% nucleotides are different. So when nucleotides will change sequence, the amino acid sequence will change. So the structure of protein will be different for different genes or amylase from different organisms. Then their localization. For example, this gene is present in cellular or chromosomal. कुठं आहे? Then clinical effects of mutation. If there is some mutation, we can do how we can do mutation. We can delete some nucleotides and add some nucleotides. Then what protein structure we'll get? Whether the activity of enzyme is as usual or it has changed, that we can understand. So for this single amylase, we can get this much information. Then also we can compare similarities of biological sequences and their structures. So what will happen? See, the amylase at different enzyme, different temperature. It will just contain active sites as usual, but little bit protein sequence is different. So this will lead to the protection. Its efficiency at low temperature, medium temperature, and high temperature. So these will be contained in the if you consider this introduction or principle, it will contain fast increase in biological information. Why there was need to develop these biological databases? Because in life sciences, thousands of people are working on a single piece of object or a research area. So the information generated is very high. So there is need to document this and convert it into databases. Then biological sciences have now turned into a data rich science. Then gene sequencing, there is the scientists are doing whole genome sequencing, then specific gene sequences. Day by day, the quantum of this work is going on increasing. Then amino acid sequences of protein are performed. Motifs and domains in protein, their sequencing is done. Structural data from XRT and NMR, it is done. Then metabolic pathway, they are operating. Then protein-protein interaction is going on. And gene expression data, microarray studies are leading to high level of information or the quantum is going on increasing in quantity. So there is a need for biological database. Now data type, there are sequence databases. First is nucleotide sequence database. Example is GeneBank, EMBL. Then protein databases are there, Swissport, PR. Then there is structural database, PDB, NDB, DAL, MSD. Similarly, enzyme, pathway, disease, literature, microarray. So if you want to look for nucleotide sequence, you will visit these sites, GeneBank or EMBL. If you want literature database, you'll go for PubMed or Scopus. So in this matter, it works. Now the main objective of database is what? Recognize various data formats and know what their primary use is there. So if you are working on a gene amylase, I've given you example. You have did a sequencing of amylase from E. coli. So sequence information what? Nucleotide. So where you should go? In nucleotide database. Then to know and understand and utilize all types of sequences identifiers. Then know and understand various features present in the gene bank flat files. And know and understand various gene bank divisions. So here main objective is databases. What features it will have? If you compare which database is good and which is not as good as this database, you have to see these features. Data heterogeneity means maximum diversity of data. Then high volume data. For amylase gene, a database, for example NCBI, it will contain thousands of base pairs, thousands of sequences. So high volume of data for single amylase gene. Then uncertainty it should have. Then data curation is also important. Large scale data integration is important. We can compare thousands of base pair at a time. So integration is very important. It is easy to share this data, and dynamic and subject to change. As the science will progress, whatever changes are made, it should be corrected in these their databases. So these features should be present in biological database. Now we come to classification. Now sequence databases. structural database and functional database. They are classified in three types. Sequence database will contain nucleic and nucleic acid and protein sequences stored in sequence database. Structural database will contain structures of RNA and protein. And functional database, the name itself suggests whatever gene is there or protein is there, what function it is carried on. It is physiological role of that gene. For example, enzyme activity, mutant phenotypes, biological pathways. Then model organism database are functional databases that provide species specific data. Model organism database, for example, E. coli database. In this database, they will contain 3,000 genes of sequence nucleotide sequences of obtained from E. coli genome. Then they will also contain function of these genes, structure, protein structure, etc. So these are model organism databases. Now we come to protein sequence databases. Now a protein database is one or more database about proteins, which could include a protein amino acid sequence, conformation, structure, and features such as active site. So, what a protein database will compose of? It is one or more data sets about proteins. So, everything we here will get about proteins only. But if we see the diverse area of protein research, what will it contain? Protein amino acid sequence, methionine, guanine. Sorry, methionine is there. Then whatever protein amino acids are there, 21 amino acids. त्यांचं sequence असेल. Then their confrontation, how in which form they are present. Then structure, what is the three-dimensional structure, and features such as active site. In an enzyme, if there contain 41 proteins, out of this, which part of protein acid sequence is composed of the active site? And active site will carry these biological activities. So protein databases are compiled by translation of DNA sequences from different gene databases. So how they are made? Nucleotide sequence is taken from nucleotide. Three pair of three nucleotides will form an amino acid, and they will form an amino acid. And from this amino acid, we can get a primary protein chain. This primary protein chain is subjected to structural databases, so we can know the structural information. Why they are so important? Every biological activity is done by protein, whether it may be growth, metabolism, reproduction, everything. So these biological data functions are performed by proteins only. So it is urgent need to maintain protein databases. Now what are the importance of protein databases? Huge amount of data for protein structure, function, and particular sequences are being generated. Now searching databases are often the first step in the study of new protein. It has following uses. For example, see, amylase enzymes आपण परत example घेऊ. So if I'm doing, so we are doing research on amylase from E. coli and we have published the paper. Other person is doing amylase from humans. Some other person are doing amylase from bacteria. So they are published in various journals. So if I want sequence of this amylase gene, then what I have to do? I have to search in Google for people working on amylase. Then I have to click their names. From their names, I have to see where which journals they have published. So a single person he is working on amylase from bacillus group of microorganisms. So he is working on 20 different bacillus. So I have to isolate or retrieve 20 research paper published on that bacillus. From that research paper, I have to copy the sequence. This is one part. Then for humans, I have to do same thing. So for only data collection, it will take 6 months. Then again, I have to put that in Excel sheet. Then again, I have to go for comparative. Structureचं लांबचंच आहे, फक्त only we are talking about sequences. So how difficult this work would be? Instead of it, what happened? I've been working on protein database. I got novel protein. I have submitted this to the database. What database will take? It will take my protein data sequence and incorporate it into their collection. Means next person, for example, there are 1,000 sequence of nucleotide for amylase gene. I have added from myself one, so 1,001. So next researcher will get 1,001 sequences at a time in a single click. So the ease of retrieving information is very simple here. Then comparison between protein or between protein families provide information about the relationship between proteins within a genome or across different species, and hence offer much more information that can be obtained by studying only an isolated protein. So if you are, again I'll give example of amylase protein. So the amylase protein which is present in bacillus, they will have close relationship in their genome. And then again, amylase gene which is present in bacillus and E. coli, they will also have close resemblance. But amylase gene in human and bacteria, it will be different little bit. The difference is higher in the form of different inclusion or deletion of amino acids. So that we can get this information by obtaining or studying only an isolated protein. Then secondary database derived from experimental databases are also widely available. These databases recognize and annotate the data or provide predictions. म्हणजे हे काय असणार आहे? If we are converting this primary sequence into the 3D structure of that protein, it is only a prediction. Prediction कसा असेल? They have designed the computer programs in which the environment of water is there and various pH or regarding other things are present in that artificially, and in that this we put our sequence, so we can get 3D structure. Everything is virtual, and virtually you are getting the structure. So we are saying this is they provide the predictions. In reality, if you want the structure, what you have to do, you have to isolate this amylase enzyme, that protein, and then you have to subject for XRD and NMR, and from that you can get the proper structure. But here in bioinformatics software, we are doing it virtually. Then use of multiple database often helps researcher understand the structure and function of protein. So at a time you can use primary protein database, multiple, they are interconnected, they share their data. Every time if I am submitting amylase gene to one protein database, the single A protein database will share it with B protein database. So there is sharing of information. I have to not submit to 6 different or 7 different protein databases. Once the information is uploaded, it is available universally. Now this is introduction about protein databases. So they are divided into two types, primary and secondary. जर तुम्हाला नोट आली, हॅलो राजकोर,
हा सर.
हा सगळ्यांना आवाज येतोय ना नीट?
हो हो.
If there is a note, see, for example, 10 mark question, what are protein, what are databases, and give example of explanation of protein sequence databases. Now you have to write for definition of databases, then what are protein databases, there you have to write, and then types. Long answer ला काय येईल, हे येऊ शकतं. But for short answer, short note, they will ask describe primary sequence, any two primary sequence database, or any two secondary protein sequence database. Then you have to describe different things, or specifically they may ask describe Swissport or PIR. Then you have to write a little bit of databases, then what are protein databases and how this PIR or Swiss port works, characteristics of this. अशा पद्धतीने लिहायला लागेल तुम्हाला. So what are primary protein database? The primary name itself studies only a sequence. So primary databases hold a experimentally determined protein sequence inferred from the conceptual translation of nucleotide sequence. So what they will do, they will take a nucleotide sequence and convert it to amino acid sequence. So whatever sequence will get, the chain of amino acid, it is called as primary database. This of course is not experimentally derived information. Every time it is not experimentally derived. आपण काय अज्युम करतो की एकदा मी amylase gene isolate केला and I have sequenced that gene amylase. Then what I can do, I can convert that amylase gene sequence into a protein 3D structure. हे व्हर्च्युअली करू शकतो. एक्सपेरिमेंटली करायचं असेल तर काय करायला लागेल मला? I have to go for isolation of amylase enzyme, pure protein, and then I have to go for XRD or NMR. So it will take 1 or 2 years to do all those things. But here what I do it, only I will do PCR, forward reverse primer, specific for this amylase gene, and I will do sequencing of that. So this is not experimentally derived information. Experimentally म्हणजे wet lab experiment. Here experimentally means wet lab experimentation. Bioinformatics is a soft lab experimentation. Wet lab मध्ये आपण pipette, burette, आपल्याला फिजिकली ते दिसतं सगळं. Protein we can see in the by running it on a gel electrophoresis. But here in bioinformatics we are doing virtual experimentation. So the name soft lab. But there has a arisen as a result of interpretation of nucleotide sequence information and consequently must be treated as a potentially containing misinterpreted information. So we have derived this interpretation of nucleotide sequence. So sometimes it can be misinterpreted also. For example, while submitting this sequence, some changes are being made for me from me. एक जरी न्यूक्लियोटाइड चेंज झालं तरी तुमचं पूर्ण अमायनो ऍसिडचं एक्सप्रेशन चेंज होतं. तर काय काय मिसइंटरप्रिटेशन पण होऊ शकतं. There is number of primary protein sequence database and each requires some specific consideration. So we'll study the different types of primary databases present. So for your syllabus, you have got Swissport, PIR, MIPS, NRL 3D, and TREMBL. These 5 types of primary protein sequence databases are present. So first is PIR. Full form stands for Protein Sequence Database. So PIR PhD is a collaborative endeavor between PIR, the MIPS, Munich Information Center for Protein Sequence, Germany, and JIPID, Japan International Proteins Information Database. So यांच्या कोलॅबोरेटिव्ह ह्याच्यामध्ये हे PIR PhD एस्टॅब्लिश झालेला आहे. So this PIR PhD is now a comprehensive, non-reductant, expertly annotated, object-related database management system. DBMS means database management system. Now, what unique characteristics it has? PIR PhD is its classification of protein sequence based on the superfamily concept. The proteins will have different classification as enzymes they will have. So proper classification of proteins is done, and in this classification there are superfamilies, and under that we'll get your required protein. So properly this arrangement of data is done in this sequence. Sequence database मध्ये proper arrangement based on classification of superfamily concept is done. So the sequences which are present in PIR PhD is also classified based on homology domain. Homology means similarity between various domain, and sequence motifs. Then homology domains may correspond to evolutionary building blocks. Evolutionary building blocks means during evolution how, for example, amylase enzyme पहिल्यांदा इकोलामध्ये होतो, नंतर तो बॅक्टेरिया डेव्हलप झाल्यानंतर कुठले झाले, पॅरामोशियम डेव्हलप झाले. These are zooplankton, then phytoplankton, then again land-based plants and animals they started developing, amphibians started. मग त्यांच्यामध्ये हे इव्होल्युशन कसं झालं आम्हाला जीनचं, that also you can study evolutionary building blocks, while sequence motif represent the functional sites. Sequence motifs म्हणजे काय, these are sequence present within the sequence, so these are functional sites or conserved region. फंक्शनल साइटचं काम काय, दे विल फ्रॉम द ऍक्टिव्ह साइट. अँड कॉन्झर्व रिजन म्हणजे हा, कॉन्झर्व रिजन म्हणजे व्हॉट एव्हर द अमायनो ऍसिड सिक्वेन्स इज देअर, इट इज हायली कॉन्झर्व. म्हणजे फॉर एक्झाम्पल 100 अमायनो ऍसिडचा सिक्वेन्स आहे रिप्रेझेंटिंग एक्स एन्झाइम और प्रोटीन, सो आउट ऑफ दॅट दिस 20 or 30 अमायनो ऍसिड्स दे विल बी कॉन्झर्व रिजन. कॉन्झर्व रिजन म्हणजे काय असणार आहे, ते कुठेतरी मध्ये आधी असणार आहे, which will form the active site, and for forming the active sites the exact sequence should repeat. आणि नॉन कॉन्झर्व्ह म्हणजे काय, बाकीचं आहे सपोर्टिंग अमायनो ऍसिड आहे, जे स्ट्रक्चर हे एक्स वाय झेड ला सपोर्ट करेल. म्हणून तर अमायलेज मध्ये काय असणार आहे, अमायलेज फ्रॉम इकोलाय आणि अमायलेज फ्रॉम ह्युमन, कॉन्झर्व्ह रिजन विल हॅव्हिंग 99% सिमिलॅरिटी, बट नॉन कॉन्झर्व्ह रिजन विल हॅव 60, 70, 40% सिमिलॅरिटी ओनली. हे आलं लक्षात, कॉन्झर्व्ड अँड नॉन कॉन्झर्व्ड रिजन. इफ दिस फंक्शनल साइट और कॉन्झर्व्ड रिजन मध्ये काय म्युटेशन झालं, देन देर इज लॉस ऑफ द ऍक्टिव्हिटी. आता मेलेनिन आपण बघितले ना, काय काय लोकांना कोड असतो. तो व्हाय दिस डिसीज इज कॉज्ड, बिकॉज देर इज नो प्रॉपर मेलॅनिन सिंथेसिस. का होत नसेल, सिक्वेन्स तर असेल पण कॉन्झर्व्ड और फंक्शनल साइट मध्येच काहीतरी म्युटेशन झालं असेल, त्यामुळे ते मेलॅनिन तयारच होत नसेल. Whenever there is loss or differences in this functional site, the protein will lose its activity. And last is the classification approach allows a more complete understanding of sequence, function and structure relationship. The sequence function and structure काय असेल, तर काय बाबा, फॉर एक्झाम्पल कार्बोहायड्रेट डिग्रेडिंग एन्झाइम्स असतील तर कसे ते क्लोजली रिलेटेड आहे, प्रोटिओलायटिक एन्झाइम्स असतील ते कसे क्लोजली रिलेटेड आहे, हे ग्रुपमध्ये आपल्याला मिळू शकतं. So this is PIR, protein sequence database. Then second is very famous, that is Swissport. Now this Swissport, like the PIR PhD, is curated protein sequence database, also provide a high level of annotations. The data in each entry can be considered separately as core data and annotation. Means there is every, for single entry there is a separate file is present here. The core data consists of sequence entered in a common single letter amino acid code. So what we'll have, various amino acid 20 amino acid असेल त्यांच्या पॅटर्न प्रमाणे तो अमायनो ऍसिडचा कोड असणार. आपल्याला दिसताना काय दिसणार, तो कोड दिसणार. जसं आपल्या न्यूक्लियोटाइडचं ATGC असतं, दिस वी कॅन सी इन द सिमिलर पॅटर्न हियर यू कॅन सी द अमायनो ऍसिड कोड and the related reference or bibliography. For example, we are working on amylase enzyme, we are from E. coli, so we have submitted this nucleotide sequence. From this nucleotide sequence they will devise the amino acid code. मग दिसताना काय दिसणार, हा ऑथर कुणी हा एक्सट्रॅक्ट केलेला आहे त्या ऑथरचे नाव, अमायनो ऍसिड कोड, देन रेफरन्स वेअर दे हॅव पब्लिश्ड, व्हॉट इज द बिब्लिओग्राफी डाटा, हे सगळं तुम्हाला स्विस्पोर्ट मध्ये दिसतं. The taxonomy of the organism from which the sequence was obtained also forms part of this core information. Taxonomy means E. coli. If you consider E. coli, it will come under microorganisms, bacteria. Gram negative, gram positive, जे काय taxonomy असेल तर ते दिसणार. If you are working on a plant, a tobacco, so taxonomy of that plant will also be shown. Genus, species, under which family it comes. हे सगळे इन्फॉर्मेशन तुम्हाला स्विस्पोर्ट मध्ये मिळेल. Now the annotation contains information on function or functions of the protein. काय काय तुम्हाला इन्फॉर्मेशन मिळेल, what is the specific function or function of that protein. Then post translational modification such as phosphorylation. जेव्हा प्रोटीन काय होतं, once the protein is primary protein is synthesized, it goes under post translational modification. तेव्हाच ते ऍक्टिव्ह प्रोटीन होऊ शकतं. ऍक्टिव्ह एन्झाइम इट कॅन परफॉर्म वेन इट विल गो अंडर पोस्ट ट्रान्सलेशनल मॉडिफिकेशन. नुसतं तुम्हाला प्रोटीन सिक्वेन्स बघून कळणार नाही ना, पोस्ट ट्रान्सलेशनल मॉडिफिकेशन काय होतंय, दॅट डाटा ऑल्सो यू कॅन गेट as phosphorylation, acylation, etc. Then functional and structural domains and sites. त्यातला कॉन्झर्ड फंक्शनल स्ट्रक्चर डोमेन्स, कॉन्झर्ड रिजन और साइट कुठली आहे, दॅट ऑल्सो यू कॅन नो, such as calcium binding site, ATP binding site, zinc finger motifs, etc. From this database also you can know the secondary structure features, as for example alpha helix, beta sheets. The protein काय असतं, सिक्वेन्स फॉर्म मध्ये इथं पडलेला नसतो. इन वॉटर इट विल फॉर्म 3 डायमेन्शन स्ट्रक्चर, अल्फा हेलिक्स, बीटा शीट्स. तर कशा फॉर्म मध्ये तो प्रेझेंट आहे ते पण, दॅट ऑल्सो डाटा यू कॅन गेट इन दिस स्विच स्पोर्ट. द quaternary structure of that protein, primary, secondary, tertiary and quaternary, that also you can know. Similarities to other protein if any, and diseases, this protein is closely related to which protein, that also you can get. And diseases that may arise due to different authors publishing different sequences from the same protein, or due to mutation in different strains of a, and described as part of annotation. So for example, mutation कसं करणार, if you want to cause physical mutation, what you have to do, you will expose it under UV light. UV light मध्ये expose झाल्यानंतर काय होईल, DNA will get damaged. Once the DNA is damaged, there will be addition deletion of these nucleotides, so there will be mutation. म्युटेशन झाल्यानंतर अमायलेज, आय विल टेक एक्झाम्पल अगेन, अमायलेज प्रोटीन मध्ये काय काय चेंजेस होतात, ऍक्टिव्हिटी इन्क्रीज होती, डिक्रीज होती, स्टॉप होती, कुठल्या ऑथरनी ते केलेलं आहे, हा डाटा मिळेल. बट व्हॉट वी कॅन डू, artificial also we can induce mutation. What you have to do, we have to add some amino acid, remove some amino acid and put that sequence for algorithmic computational programs. So there you can get the three-dimensional structure. There you can study whatever the function has changed. Physical data also, soft data also, or wet lab data also. That also you will get in Swissport. Now, baga, see in a single Swissport how many parameters of protein they have covered. And ha sagala data at a single click you will get. So this is a very famous database, Swissport. Hello? Mansi? राजकोर?
हाँ सर बोला.